Okay, good afternoon. Thanks for Murayama introduction. I'm very happy here at FHS to have this opportunity to share our studies with all of you. I'm from Department of Clinical Laboratory Sciences and Medical Biotechnology, College of Medicine, National Taiwan University. My lab has been working on a uropathogen called Proteus mirabilis. So today's my topic is a copper detoxification system in uropathogenic Proteus mirabilis. There are three reasons for this study. The first is because UTI is urinary tract infection is a very important infection. And second, the Proteus mirabilis is a frequent uropathogen. And third, is the copper concentration will increase during UTI. So let me first introduce the bacteria. The bacteria is a gram-negative facultative anaerobic belongs to Enterobacteriaceae. Because UTI, especially coccidia-associated, so-called COTI, it can also cause other infections such as wound infection, septicemia, and pneumonia. The bacteria has several important virus factors, including flagella for motility and hemolyzing to damage the urine epithelial cells, and the ureals to degrade the urea in the urine to make the urine alkaline to facilitate the formation of urine stone. And also IgA Proteus to counteract the human host immune response, and also fibrin for the bacteria to attach to the epithelial cell to avoid the urine flow, and also facilitate the formation of biofilm. And as a very interesting, the bacteria has a very special form of motility called swarming. Swarming is a motility on the solid medium, as different from that of swimming is in the liquid medium. So we can see here the swarming is a cyclic process involving differentiation of single rod swimming cell into elongated multicellular swarmer cell, and to start migration on a solid medium, and differentiation from swarmer cells back into the planktonic swimming cells. It's a process also called consolidation. So when we inoculate a drop of bacterial suspension onto the center of the agar surface of LB with 1.5 to 2% agar, a bullseye swarming pattern can be seen, like this figure shows. This is not a cartoon. And there are several strategies for uropathogen to establish a UTI. First, adhere against the urine flow, and second, resistant to innate immunity, including antimicrobial peptides and immunoglobulins. And they also possess several stress response mechanisms to cope with nutrient deficiency and attack of antibiotics and reactive oxygen species. They also regulate the gene expression to make sure to express the right gene at the right time. On the other hand, as we know, copper is an essential nutrient but toxic when in excess. And copper has very important biological roles. For example, a catalytic role as an enzyme cofactor and for protein structural maintenance. The copper toxicity from oxidative damage from Fenton reaction, shown here. The cupric ion can react with the hydrogen peroxide to produce the highly toxic hydroxy free radical to damage the macromolecules in our body. And then it can cause protein misfolding and aggregation or disrupt the protein with iron sulfur cluster, which are important members in respiration and metabolic processes. And copper also is a member of innate immunity. During infection, the copper will imported into the macrophage and into the phagosome to help to kill the bacteria. And several studies indicate copper is a host effector mobilized to urine during urinary tract infection to impel bacterial colonization. The left diagram shows when upon infection by UPEC, uropathogenic E. coli, or Klebsiella pneumonia or Proteus mirabilis, the urine copper concentration will increase dramatically compared to the healthy control. Okay, and then the right panel shows that the copper deficient will promote UPEC colonization in mice. So this we can see the white bar is the copper deficient mice, and the white is control. And we can see both in bladders and kidney, the copper deficient mice, the UPEC has higher colonization rate. Okay. So we wonder what is the interaction between copper and the Proteus mirabilis during the infection. So we perform transposon mutagenesis to isolate copper-related mutants. Copper-related mutant and at the concentration of copper 3 millimolarity, at which the wild type can survive, but not the mutants. And several mutants were isolated, and we identify the genes disrupted by the transposon and individual mutant, as this show the COP-A and the QR and the transposon insertion site here. We then perform gene knockout by homologous recombination to confirm the transposon mutant. So we first select 3 mutants, QO, QR, and COP-A for further study. We compare the amino acid of QO, QR, and COP-A or Proteus mirabilis to the counterparts of UPEC, and then they all show high similarity to those of UPEC. You can see here the over 70% similarity. So as we know, copper resistant mechanisms are different among different bacteria. Q system is one of them. in E. coli, the Q systems consist of three members, QO, QR, and COP-A. When the cuprate ion the concentration of cuprate ion increase inside the cytoplasm, the ion will combine with the QR. The QR is the copper sensing transcription factor, and then trigger the downstream gene expression of COP-A and QO. Now, COP-A is a copper efflux pump. It will pump out the toxic cuprate ion, and then into the periplasm, and in this phase, the QO is a multi-copper oxidase will conversion the more toxic cuprate ion into the less toxic cupric ion. So to release the stress of copper exposure. So firstly, we confirm the copper susceptibilities of these isolated mutants. And we can see all the mutants include HHA and QR, COP-A and QO. All the mutants exhibit decreased survival upon exposure to copper of 2.5 millimolarity. Because QR is a transcription factor to control the expression of QO and COP-A expression at the transcriptional level, we search the QR consensus binding site in the promoter region of QO and COP-A and Proteus mirabilis. And we did find the consensus QR binding sequence binding site shown here as in E. coli and Salmonella type murine. And more and more data research have indicate that copper resistance is related to is highly associated with bacterial virulence. So this is a paper published in Frontier Microbiology. They characterize the copper resistant adherence and of Acinetobacter baumannii. So we firstly analyze the QO mutant. We want to see the effect of QO deletion on virus vector expression, including biofilm formation, urease activity, and urine stone formation. As we can see here, the biofilm formation, the three phenotype were not affected by the mutation of QO. This is a wild type, this is a mutant. And next we evaluate the QO deletion on tolerance to hydrogen peroxide and the survival co-culture with macrophages. The left diagram shows QO has a decreased survival rate on exposure to 20 millimolarity hydrogen peroxide, and also has the lower survival rate co-culture with macrophage. The two data are parallel because we know the macrophage is an environment rich in oxidative stress like hydrogen peroxide. And then we further analyze the ability of adhesion and the invention into urothelium cell of the QO mutant. Then we can see the QO mutant it has a lower adhesion and the invention abilities compared to the wild type. Then we also evaluate the cytokine expression of urocilin cells by QO mutation. As we can see in the figure that the QO mutant has the highest TNF alpha, interleukin 8 and interleukin 6 messenger RNA label compared to the wild type and no bacterial control. This result led us hypothesize that the mutant may have a in vivo virus. Next we perform mouse colonization assays using QO mutant and wild type strain. And in this figure, we can see QO mutant has significantly lower colonization ability in the bladder, in the mouse bladder. And in a similar way, the QR mutant also has lower colonization ability than the wild type, both in bladder and kidneys of mice. And when we supplement the copper into the drinking water of mice, we can observe that the colonization rate is further lower in both wild type bacteria and QR mutant, both in bladder and kidneys. So we conclude that loss of QR or QO impairs colonization in mice. And another is copper supplementation reduces Proteus mirabilis colonization. Yeah, sorry. So I cannot, okay. Yeah. And then we wonder if copper can induce CoB and the QO expression to release the copper stress in Proteus mirabilis. So we perform RT-qPCR to measure the messenger RNA level of CoB and QO in the presence of 1 millimolar copper control to the LV only control. And we can see here the CoB express the messenger RNA level and the QO increased dramatically compared to the control. And then we demonstrated the copper induced expression of QO and CoB is dependent on the presence of QR transcription factor. So we can see here the QO promoter activity and promoter of CoB was induced by copper here and here, but not in the QR mutant. So we conclude that the copper induced QO and CoB depends on the presence of QR transcription factor. So in conclusion, firstly, for the first time, we demonstrate a copper detoxification system in Proteus mirabilis. And second, copper binding QR can activate expression of a QO and CoB to confer copper resistance. And third, both QR and QO contribute to virulence. And fourth, the QR and the QO could be the candidates of drug target for prevention of UTI caused by Proteus mirabilis. Okay, thank you for your attention, and I want to say this is my first time to attend the FHS conference, and I hope you can learn something from my talk. Okay, thank you.
Thank you very much for your excellent presentation, Dr. Liaw.